So hello, everyone. I've been given the task of giving an update on the development atlas work. I'll be giving an update, but mainly as vignettes. So it's a little bit of a birthday for the development work as well in terms of the Human Cell Atlas, because we had our official sort of scheduled session last year. And at that session, it was much more about what we were going to do and the potential. So now, this year, we can actually present some of the more concrete data that we've got, some of the kind of like issues that have arisen, and also how we plan to move forwards. The first thing to say is the Human Cell Atlas initiative has really catapulted the study of human development into the limelight because of the technologies that have been made possible to be used in terms of studying human development. And this really has been a fantastic opportunity because for once, we actually have real time in studying human development and also across many tissues from one sample, which is a very unique sort of opportunity and scientific endeavor. And as with any of these sort of atlas approach, particularly in terms of studying human development, having good resources is very important. And there is one such resource, which I will mention, which is the Human Developmental Biology resource, having been funded by Wellcome and MRC for the last 18 years, and not just serves the community, research communities in the United Kingdom, but also worldwide. And many, including people who have undertaken some of the work that I will show, have benefited from this resource. But there are also many other resources that are available. So what I'm going to do now is to show some of the efforts across the community. And I'll start with one which is sort of closer to home, which is really work that has been funded by Wellcome. And the focus here has been looking at the fetal-maternal interface, but also the developing blood and immune systems. And just to highlight two kind of main areas. One is work that maybe you've heard a little bit earlier with looking at the deciduant placenta, where you have the cohabitation of maternal cells and also fetal cells, how you can use these single-cell technologies, not just RNA sequencing, but also DNA sequencing to work out what are the maternal cells and fetal cells which are existing in this sort of harmonious environment to actually tolerate the fetus and ensure a successful pregnancy. And the second is some of the kind of like earlier works to come out from the studies of the developing blood and immune system, which is focusing on the liver, which is one of the major sites of hematopoiesis during development. And we're very good at sort of deconstructing tissues and identifying sort of single cells that are within there. But I think we've also now come to the an area or stage where we're now wanting to look at them in situ. And I think Sarah's mentioned a lot of the methods and technological advances that we can do that. But also in terms of trying to understand how these cells are actually communicating with one another. And allied with this work is a exploitation of a database that will allow you to look at how cells are communicating, what are the receptor-ligand relationships, so one could computationally reconstruct the tissue. But I think, nevertheless, some of the challenges that still remain, which is where you have a transcriptome-based cell clusters from which you now have to derive a way of tangibly isolating the cells, validating their identity, and then taking it to the next step in terms of studying what they look like, what their functions are, and so on and so forth. And that's still not straightforward because at this point in time, by and large, what we do is try and find markers that we can use to identify the clusters, isolate the cells, validate that they do indeed match to the original cells. And some of this pioneering work, which wasn't part of the Atlas initiative, but was looking at the peripheral blood dendritic cells and monocytes, which was led by Chloe Villani and Nir Hakohan, where we really tried to look at the populations and what they functionally identified as, and not just as a cluster by transcriptome. So those are still challenges that remain. Now looking at some of the efforts that are coming from Sweden, where there is a slightly parallel and orthogonal method in terms of correlating the transcriptome-based cell identities with space. And here, it's a parallel approach where you do the dissociation, but you can also look at the spatial information by looking at the transcriptome of regions using spatial transcriptomics, and then integrating of how the dissociated cells match up to where they are, and you can subsequently zoom in further by elaborating on smaller regions and looking at the transcriptome. And here and another way of doing this, which is with Matt Nilsson's and Joachim Lundeberg, is where combining spatial transcriptomics and looking at specific areas of interest, and then doing in situ sequencing of several hundred transcripts at the same time. So these are new approaches that we hope to sort of exploit further in the study of human development. And then I'm going to sort of finish off with the work that's been undertaken by Arnold Kriegstein in the US, which is looking at how the human brain develops, and not just in terms of what are the cell types that are in there, but also functional sort of identities of those cells. And as I mentioned, this is by no means a comprehensive account of all of the Development Atlas projects that are going on around the world. For a start, I've not mentioned some of the work in kidney that Sam Bejati has been leading, but also recent publications on kidney and such. But I wanted to highlight one of the kind of I think, critical discussion points, which is essentially relating to annotation and nomenclature. And I very much welcome this kind of like a call from the analysis computational groups. How do we annotate these sort of fetal cells? And I think particularly in the immune system, we have basically a situation where Cambridge, Massachusetts is different from Cambridge, UK, but they're both called Cambridge. So how do we make more kind of a relevance? How do we really understand it? Because some of these markers that faithfully mark adult cells are not reliable during development. So I think a lot of these issues need to be talked about. But I think one good thing that's coming out of all of this is that the developmental atlas research community really want to work together. We're trying to sort of spearhead a coordinated platform in terms of the methods, the technological methods that we're using, complementary organs dissection, complementary expertise, and having a sort of a shared website and communication across the groups. And that's really what we want to push forward. So maybe we'll give an update on that next year in terms of how we're progressing. Oh, it's red. Thanks.